practice what you've learned and reinforce. Carbohydrates are the most abundant biomolecules on Earth. Each year, photosynthesis converts more than 100 billion metric tons of CO2 and H2O into cellulose and other plant products. Certain carbohydrates, sugar and starch, are a dietary staple in most parts of the world, and the oxidation of carbohydrates is the central energy-yielding pathway in most non-photosynthetic cells. Carbohydrate polymers, also called glycans, serve as structural and protective elements in the cell walls of bacteria and plants, and in the connective tissues of animals. Other carbohydrate polymers lubricate skeletal joints, and participate in cell-cell recognition and adhesion. Complex carbohydrate polymers covalently attached to proteins or lipids act as signals that determine the intracellular destination or metabolic fate of these hybrid molecules, called glycoconjugates. This chapter introduces the major classes of carbohydrates and glycoconjugates and provides a few examples of their many structural and functional roles. Carbohydrates are polyhydroxyaldehydes or ketones, or substances that yield such compounds on hydrolysis. Many, but not all, carbohydrates have the empirical formula CH2ON. Some also contain nitrogen, phosphorus, or sulfur. There are three major size classes of carbohydrates: monosaccharides, oligosaccharides, and polysaccharides. The word saccharide is derived from the Greek saccharin, meaning sugar. Monosaccharides, or simple sugars, consist of a single polyhydroxyaldehyde or ketone unit. The most abundant monosaccharide in nature is the 6-carbon sugar D-glucose, sometimes referred to as dextrose. Monosaccharides of 4 or more carbons tend to have cyclic structures. Oligosaccharides consist of short chains of monosaccharide units, or residues, joined by characteristic linkages called glycosidic bonds. The most abundant are the disaccharides, with 2 monosaccharide units. Sucrose. Cane sugar, for example, consists of the 6-carbon sugars D-glucose and D-fructose. All common monosaccharides and disaccharides have names ending with the suffix -ose. In cells, most oligosaccharides consisting of 3 or more units do not occur as free entities, but are joined to non-sugar molecules, lipids, or proteins, in glycoconjugates. The polysaccharides are sugar polymers containing more than 20 or so monosaccharide units. Some have hundreds or thousands of units. Some polysaccharides, such as cellulose, are linear chains. Others, such as glycogen, are branched. Both cellulose and glycogen consist of recurring units of D-glucose, but they differ in the type of glycosidic linkage and consequently have strikingly different properties and biological roles. 7.1 Monosaccharides and disaccharides. The simplest of the carbohydrates, the monosaccharides, are either aldehydes or ketones with 2 or more hydroxyl groups. The 6-carbon monosaccharides glucose and fructose have 5 hydroxyl groups. Many of the carbon atoms to which the hydroxyl groups are attached are chiral centers, which give rise to the many sugar stereoisomers found in nature. Stereoisomerism in sugars is biologically significant because the enzymes that act on sugars are strictly stereospecific, typically preferring one stereoisomer to another by 3 or more orders of magnitude, as reflected in Km values or binding constants. It is as difficult to fit the wrong sugar stereoisomer into an enzyme's binding site as it is to put your left glove on your right hand. Figure 6. We begin by describing the families of monosaccharides with backbones of 3 to 7 carbons, their structure, their stereoisomeric forms, and the means of representing their three-dimensional structures on paper. We then discuss several chemical reactions of the carbonyl groups of monosaccharides. One such reaction, the addition of a hydroxyl group from within the same molecule, generates cyclic forms with 4 or more backbone carbons, the forms that predominate in aqueous solution. This ring closure creates a new chiral center, adding further stereochemical complexity to this class of compounds. The nomenclature for unambiguously specifying the configuration about each carbon atom in a cyclic form and the means of representing these structures on paper are described in some detail. This information will be useful as we discuss the metabolism of monosaccharides in Part 2. We also introduce here some important monosaccharide derivatives encountered in later chapters. The two families of monosaccharides are aldoses and ketoses. Monosaccharides are colorless, crystalline solids that are freely soluble in water but insoluble in nonpolar solvents. Most have a sweet taste, see box 7.2. The backbones of common monosaccharides are unbranched carbon chains in which all the carbon atoms are linked by single bonds. In this open chain form, one of the carbon atoms is double bonded to an oxygen atom to form a carbonyl group. Each of the other carbon atoms has a hydroxyl group. If the carbonyl group is at an end of the carbon chain, that is, in an aldehyde group, the monosaccharide is an aldose. If the carbonyl group is at any other position, in a ketone group, the monosaccharide is a ketose. The simplest monosaccharides are the two 3-carbon trioses, glyceraldehyde, an aldotriose, and dihydroxyacetone, a ketotriose, Fig. 7-1a. Monosaccharides with 4, 5, 6, and 7 carbon atoms in their backbones are called, respectively, tetroses, pentoses, hexoses, and heptoses. There are aldoses and ketoses of each of these chain lengths, aldotetroses and ketotetroses, aldopentoses and ketopentoses, and so on. The hexoses, which include the aldohexose D-glucose and the ketohexose D-fructose, Fig. 7-1b, are the most common monosaccharides in nature, the products of photosynthesis and key intermediates in the central energy-yielding reaction sequence in most organisms. The aldopentoses D-ribose and 2-deoxy-D-ribose, Fig. 7-1c, are components of nucleotides and nucleic acids, Chapter 8. Monosaccharides have asymmetric centers. All the monosaccharides except dihydroxyacetone contain one or more asymmetric, chiral, carbon atoms and thus occur in optically active isomeric forms, pages 17 to 18. The simplest aldose, glyceraldehyde, contains one chiral center, the middle carbon atom, and therefore has two different optical isomers, or enantiomers, Fig. 7-2. Key convention. One of the two enantiomers of glyceraldehyde is, by convention, designated the D isomer, the other is the L isomer. As for other biomolecules with chiral centers, the absolute configurations of sugars are known from X-ray crystallography. To represent three-dimensional sugar structures on paper, we often use Fischer projection formulas, Fig. 7-2. In these projections, horizontal bonds project out of the plane of the paper, toward the reader. Vertical bonds project behind the plane of the paper, away from the reader. Figure 7. Glyceraldehyde. Stick models that in plane of the paper. In general, a molecule with N chiral centers can have two N stereoisomers. Glyceraldehyde has 2 1 is equal to 2. The aldohexoses, with 4 chiral centers, have 2 4 is equal to 16. The stereoisomers of monosaccharides of each carbon chain length can be divided into two groups that differ in the configuration about the chiral center most distant from the carbonyl carbon. Those in which the configuration at this reference carbon is the same as that of D-glyceraldehyde are designated D isomers, and those with the same configuration as L-glyceraldehyde are L isomers. In other words, when the hydroxyl group on the reference carbon is on the right, dextro, in a projection formula that has the carbonyl carbon at the top, the sugar is the D isomer, when on the left, levo, it is the L isomer. Of the 16 possible aldohexoses, 8 are D forms and 8 are L. Most of the hexoses of living organisms are D isomers. Why D isomers? An interesting and unanswered question. Recall that all of the amino acids found in proteins are exclusively one of two possible stereoisomers, L, P, 78. The basis for this initial preference for one isomer during evolution is unknown. However, once one isomer had been selected, it was likely that evolving enzymes would retain their preference for that stereoisomer. Figure 7-3 shows the structures of the D stereoisomers of all the aldoses and ketoses having 3 to 6 carbon atoms. The carbons of a sugar are numbered beginning at the end of the chain nearest the carbonyl group. Each of the 8 D-aldohexoses, which differ in the stereochemistry at C2, C3, or C4, has its own name, D-glucose, D-galactose, D-mannose, and so forth, Fig. 7-3a. The 4 and 5 carbon ketoses are designated by inserting ol into the name of a corresponding aldose. for example, diribulose is the ketopentose corresponding to the aldopentose D-ribose. The importance of ribulose will become clear when we discuss the fixation of atmospheric CO2 by green plants in Chapter 20. The Ketohexoses are named otherwise, for example, fructose from the Latin fructus, fruit. Fruits are one source of this sugar, and sorbose, from sorbus, the genus of mountain ash, which has berries rich in the related sugar alcohol sorbitol. Two sugars that differ only in the configuration around one carbon atom are called epimers, D-glucose and D-mannose, which differ only in the stereochemistry at C2, are epimers. As are D-glucose and D-galactose, which differ at C4, Fig. 7-4. Some sugars occur naturally in their L form. Examples are L-arabinose, and the L-isomers of some sugar derivatives that are common components of glycoconjugates, section 7.3. The common monosaccharides have cyclic structures. For simplicity, we have thus far represented the structures of aldoses and ketoses as straight-chain molecules, Fig. 7-3, 7-4. In fact, in aqueous solution, aldotetroses and all monosaccharides with 5 or more carbon atoms in the backbone occur predominantly as cyclic, ring, structures, in which the carbonyl group has formed a covalent bond with the oxygen of a hydroxyl group along the chain. The formation of these ring structures is the result of a general reaction between alcohols and aldehydes or ketones to form derivatives called hemiacetyls or hemiketals. Two molecules of an alcohol can add to a carbonyl carbon. The product of the first addition is a hemiacetyl for addition to an aldose, or a hemiketal for addition to a ketose. If the OH and carbonyl groups are on the same molecule, a 5 or 6-membered ring results. Addition of the second molecule of alcohol produces the full acetal or ketal, Fig. 7-5, and the bond formed is a glycosidic linkage. When the two molecules that react are monosaccharides, the acetal or ketal formed is a disaccharide. Figure 7-3 aldoses and ketoses. The series of A, D-aldoses and B, D-ketoses having from 3 to 6 carbon atoms, shown as projection formulas. The carbon atoms in red are chiral centers. In all these D-isomers, the chiral carbon most distant from the carbonyl carbon has the same configuration as the chiral carbon in D-glyceraldehyde. The sugars named in boxes are the most common in nature. You will encounter these again in this and later chapters. The reaction with the first molecule of alcohol creates an additional chiral center, the carbonyl carbon. Because the alcohol can add in either of two ways, attacking either the front or the back of the carbonyl carbon, the reaction can produce either of two stereoisomeric configurations, denoted alpha and beta. For example, D-glucose exists in solution as an intramolecular hemiacetal in which the free hydroxyl group at C5 has reacted with the aldehydic C1, rendering the latter carbon asymmetric and producing two possible stereoisomers, designated alpha and beta, Fig. 7-6. Isomeric forms of monosaccharides that differ only in their configuration about the hemiacetal or hemiketal carbon atom are called anomers, and the carbonyl carbon atom is called the anomeric carbon. Figure 7-4 epimers. D-glucose. Figure 7-5 for 6-membered ring compounds are called pyranoses because they resemble the 6-membered ring compound pyran, Fig. 7-7. The systematic names for the two ring forms of D-glucose are therefore alpha-D-glucopyranose and beta-D-glucopyranose. Ketohexoses, such as fructose, also occur as cyclic compounds with alpha and beta anomeric forms. In these compounds, the hydroxyl group at C5, or C6, reacts with the keto group at C2 to form a furanose, or pyranose, ring containing a hemiketal linkage, Fig. 7-5. D-fructose readily forms the furanose ring, Fig. 7-7. The more common ionomer of this sugar in combined forms, or in derivatives, is beta-D-fructofuranose. Figure 7. Cyclic sugar structures are more accurately represented in Hayworth perspective formulas than in the Fischer projections commonly used for linear sugar structures. In Hayworth perspectives, the 6-membered ring is tilted to make its plane almost perpendicular to that of the paper, with the bonds closest to the reader drawn thicker than those farther away, as in Figure 7-7. Key convention, to convert the Fischer projection formula of any linear Hexose to a Hayworth perspective formula showing the molecule's cyclic structure. Draw the six-membered ring, 5 carbons, and 1 oxygen at the upper right. Number the carbons in a clockwise direction beginning with the anomeric carbon, then place the hydroxyl groups. If a hydroxyl group is to the right in the Fischer projection, it is placed pointing down, i.e., below the plane of the ring, in the Hayworth perspective. If it is to the left in the Fischer projection, it is placed pointing up, i.e., above the plane, in the Hayworth perspective. The terminal CH2OH group projects upward for the D enantiomer, downward for the L enantiomer. The hydroxyl on the anomeric carbon can point up or down. When the anomeric hydroxyl of a D-hexose is on the same side of the ring as C6, the structure is by definition beta. When it is on the opposite side from C6, the structure is alpha. Worked example. Projection to perspective. Draw the Hayworth solution. Worked example. Perspective. Sugar ice. Draw the Hayworth solution. The alpha and beta anomers of D-glucose interconvert in aqueous solution by a process called mutarotation, in which one ring form, say, the alpha anomer, opens briefly into the linear form, then closes again to produce the beta anomer, Fig. 7-6. Thus, a solution of beta-D-glucose and a solution of alpha-D-glucose eventually form identical equilibrium mixtures having identical optical properties. This mixture consists of about 1/3 alpha-D-glucose, 2/3 beta-D-glucose, and very small amounts of the linear form and the 5-membered ring, glucofuranose form. Hayworth perspective formulas like those in Figure 7-7 are commonly used to show the stereochemistry of ring forms of monosaccharides. However, the 6-membered pyranose ring is not planar, as Hayworth perspectives suggest, but tends to assume either of 2 chair conformations, Fig. 7-8. Recall from Chapter 1, pages 16-19, that 2 conformations of a molecule are interconvertible without the breakage of covalent bonds, whereas 2 configurations can be interconverted only by breaking a covalent bond. To interconvert alpha and beta configurations, the bond involving the ring oxygen atom has to be broken, but interconversion of the 2 chair forms, which are conformers, does not require bond breakage and does not change configurations at any of the ring carbons. The specific 3-dimensional structures of the monosaccharide units are important in determining the biological properties and functions of some polysaccharides, as we shall see. Figure 7-8. Organisms contain a variety of hexose derivatives. In addition to simple hexoses such as glucose, galactose, and mannose, there are many sugar derivatives in which a hydroxyl group in the parent compound is replaced with another substituent, or a carbon atom is oxidized to a carboxyl group, Fig. 7-9. In glucosamine, galactosamine, and monosamine, the hydroxyl at C2 of the parent compound is replaced with an amino group. The amino group is commonly condensed with acetic acid, as in N-acetyl glucosamine. This glucosamine derivative is part of many structural polymers, including those of the bacterial cell wall. Substitution of a hydrogen for the hydroxyl group at C6 of L-galactose or L-mannose produces L-fucose or L-rhamnose, respectively. L-fucose is found in the complex oligosaccharide components of glycoproteins and glycolipids. L-rhamnose is found in plant polysaccharides. Oxidation of the carbonyl, aldehyde, carbon of glucose to the carboxyl level produces gluconic acid, used in medicine as an innocuous counterion when administering positively charged drugs, such as quinine, or ions, such as Ca2+. Other aldoses yield other aldonic acids. Oxidation of the carbon at the other end of the carbon chain C6 of glucose, galactose, or mannose forms the corresponding uronic acid, glucuronic, galacturonic, or mannuronic acid. Both aldonic and uronic acids form stable intramolecular esters called lactones, Fig. 7-9, lower left. The sialic acids are a family of sugars with the same 9-carbon backbone. One of them, N-acetylneurominic acid, often referred to simply as sialic acid, is a derivative of N-acetylmonosamine that occurs in many glycoproteins and glycolipids on animal cell surfaces, providing sites of recognition by other cells or extracellular carbohydrate-binding proteins. The carboxylic acid. Groups of the acidic sugar derivatives are ionized at pH 7, and the compounds are therefore correctly named as the carboxylates glucuronate, galacturonate, and so forth. Figure 7 to 9. Some hexose derivatives important in the synthesis and metabolism of carbohydrates. The intermediates are very often not the sugars themselves, but their phosphorylated derivatives. Condensation of phosphoric acid with one of the hydroxyl groups of a sugar forms a phosphate ester, as in glucose 6-phosphate, Fig. 7 to 9, the first metabolite in the pathway by which most organisms oxidize glucose for energy. Sugar phosphates are relatively stable at neutral pH and bear a negative charge. One effect of sugar phosphorylation within cells is to trap the sugar inside the cell. Most cells do not have plasma membrane. Transporters for phosphorylated sugars. Phosphorylation also activates sugars for subsequent chemical transformation. Several important phosphorylated derivatives of sugars are components of nucleotides, discussed in the next chapter. Box 7. Measurement, diagnosis, treatment of glucose is a second enzyme. Leave the erythrocyte. Figure 1. The non-enzyme. Figure 2. Monosaccharides are reducing agents. Monosaccharides can be oxidized by relatively mild oxidizing agents such as cupric Cu2+ ion. The carbonyl carbon is oxidized to a carboxyl group. Glucose and other sugars capable of reducing cupric ion are called reducing sugars. The sugars are oxidized to a complex mixture of carboxylic acids. This is the basis of Felling's reaction, a semi-quantitative test for the presence of reducing sugar that for many years was used to detect and measure elevated glucose levels in people with diabetes mellitus. Today, more sensitive methods that involve an immobilized enzyme on a test strip are used. They require only a single drop of blood. Disaccharides contain a glycosidic bond. Disaccharides, such as maltose, lactose, and sucrose, consist of two monosaccharides joined covalently by an O-glycosidic bond, which is formed when a hydroxyl group of one sugar molecule, typically in its cyclic form, reacts with the anomeric carbon of the other, Fig. 7 to 10. This reaction represents the formation of an acetal from a hemiacetal, such as glucopyranose, and an alcohol, a hydroxyl group of the second sugar molecule, Fig. 7 to 5, and the resulting compound is called a glycoside. Glycosidic bonds are readily hydrolyzed by acid but resist cleavage by base. Thus disaccharides can be hydrolyzed to yield their free monosaccharide components by boiling with dilute acid. N-glycosyl bonds join the anomeric carbon of a sugar to a nitrogen atom in glycoproteins, see Fig. 7 to 30, and nucleotides, see Fig. 8 to 1. The oxidation of a sugar by cupric ion, the reaction that defines a reducing sugar, occurs only with the linear form, which exists in equilibrium with the cyclic forms. When the anomeric carbon is involved in a glycosidic bond, that is, when the compound is a full acetal or ketal, see Fig. 7 to 5, the easy interconversion of linear and cyclic forms shown in Figure 7 to 6 is prevented. Because the carbonyl carbon can be oxidized only when the sugar is in its linear form, formation of a glycosidic bond renders a sugar non-reducing. In describing disaccharides or polysaccharides, the end of a chain with a free anomeric carbon, one not involved in a glycosidic bond, is commonly called the reducing end. Figure 7 to 10. Formation of maltose. The disaccharide maltose, Fig. 7 to 10, contains two D-glucose residues joined by a glycosidic linkage between C1, the anomeric carbon, of one glucose residue, and C4 of the other. Because the disaccharide retains a free anomeric carbon, C1 of the glucose residue on the right in Fig. 7 to 10, maltose is a reducing sugar. The configuration of the anomeric carbon atom in the glycosidic linkage is alpha. The glucose residue with the free anomeric carbon is capable of existing in alpha and beta pyranose forms. Key convention, to name reducing disaccharides such as maltose unambiguously, and especially to name more complex oligosaccharides, several rules are followed. By convention, the name describes the compound written with its non-reducing end to the left, and we can build up the name in the following order. 1. Give the configuration, alpha or beta, at the anomeric carbon joining the first monosaccharide unit, on the left, to the second. 2. Name the non-reducing residue. To distinguish 5- and 6-membered ring structures, insert furano or purano into the name. 3. Indicate in parentheses the two carbon atoms joined by the glycosidic bond, with an arrow connecting the two numbers. For example, 1,4 shows that C1 of the first named sugar residue is joined to C4 of the second. 4. Name the second residue. If there is a third residue, describe the second glycosidic bond by the same conventions. To shorten the description of complex polysaccharides. 3-letter abbreviations or colored symbols for the monosaccharides are often used, as given in Table 7:1. Following this convention for naming oligosaccharides, maltose is alpha-D-glucopyranosyl-1,4-D-glucopyranose. Because most sugars encountered in this book are the D enantiomers and the pyranose form of hexoses predominates, we generally use a shortened version of the formal name of such compounds, giving the configuration of the anomeric carbon and naming the carbons joined by the glycosidic bond. In this abbreviated nomenclature, maltose is GLC, alpha-1,4, GLC. Table 7:1 symbols and abbreviations for common monosaccharides, abiquote, galactose, glucose GL, mannose man, rhamnose, ribose, glucurose, galactose, glucose, N-acetyl, N-iduron. Note: the disaccharide lactose, Fig. 7-11, which yields D-galactose and D-glucose on hydrolysis, occurs naturally in milk. The anomeric carbon of the glucose residue is available for oxidation, and thus lactose is a reducing disaccharide. Its abbreviated name is GAL, beta-1,4, GLC. The enzyme lactase absent in lactose-intolerant individuals begins the digestive process in the small intestine by splitting the beta-1,4 bond of lactose into monosaccharides, which can be absorbed from the small intestine. Lactose, like other disaccharides, is not absorbed from the small intestine, and in lactose-intolerant individuals, the undigested lactose passes into the large intestine. Here, the increased osmolarity due to dissolved lactose opposes the absorption of water from the intestine into the bloodstream, causing watery, loose stools. In addition, fermentation of the lactose by intestinal bacteria produces large volumes of CO2, which leads to the bloating, cramps, and gas associated with lactose intolerance. Figure 7-11. These are shown as name and for sucrose. Sucrose is a disaccharide of glucose and fructose. It is formed by plants but not by animals. In contrast to maltose and lactose, sucrose contains no free anomeric carbon atom. The anomeric carbons of both monosaccharide units are involved in the glycosidic bond, Fig. 7-11. Sucrose is therefore a non-reducing sugar, and its stability, its resistance to oxidation, makes it a suitable molecule for the storage and transport of energy in plants. In the abbreviated nomenclature, a double-headed arrow connects the symbols specifying the anomeric carbons and their configurations. Thus the abbreviated name of sucrose is either GLC, alpha left-right arrow 2 beta, Fro or Fro, beta 2 left-right arrow 1 alpha, GLC. Sucrose is a major intermediate product of photosynthesis. In many plants it is the principal form in which sugar is transported from the leaves to other parts of the plant body. Trehalose, GLC, alpha 1 left-right arrow 1 alpha, GLC Fig. 7-11, a disaccharide of D-glucose that, like sucrose, is a non-reducing sugar, is a major constituent of the circulating fluid, hemolymph, of insects. It serves as an energy storage compound. Lactose gives milk its sweetness, and sucrose, of course, is table sugar. Trehalose is also used commercially as a sweetener. Box 7-2 explains how humans detect sweetness, and how artificial sweeteners such as aspartame act. Summary 7.1 Monosaccharides and disaccharides. Sugars, also called saccharides, are compounds containing an aldehyde or ketone group and two or more hydroxyl groups. Monosaccharides generally contain several chiral carbons and therefore exist in a variety of stereochemical forms, which may be represented on paper as Fischer projections. Epimers are sugars that differ in configuration at only one carbon atom. Monosaccharides commonly form internal hemiacetals or hemiketals, in which the aldehyde or ketone group joins with a hydroxyl group of the same molecule, creating a cyclic structure. This can be represented as a Hayworth perspective formula. The carbon atom originally found in the aldehyde or ketone group, the anomeric carbon, can assume either of two configurations, alpha and beta, which are interconvertible by mutarotation. In the linear form of the monosaccharide, which is in equilibrium with the cyclic forms, the anomeric carbon is easily oxidized, making the compound a reducing sugar. A hydroxyl group of one monosaccharide can add to the anomeric carbon of a second monosaccharide to form an acetal called a glycoside. In this disaccharide, the glycosidic bond protects the anomeric carbon from oxidation, making it a non-reducing sugar. Oligosaccharides are short polymers of several monosaccharides joined by glycosidic bonds. At one end of the chain, the reducing end, is a monosaccharide unit with its anomeric carbon not involved in a glycosidic bond. The common nomenclature for disaccharides or oligosaccharides specifies the order of monosaccharide units, the configuration at each anomeric carbon, and the carbon atoms involved in the glycosidic linkages. Box 7-2 sugar is sweet, and so are a few other things. Sweetness is one of the 5 basic flavors that humans can taste. Fig. 1. The others are sour, bitter, salty, and umami. Sweet taste is detected by protein receptors in the plasma membranes of gustatory cells and the taste bud.